Second announcement is that the guy who really discovered the RDHV virus, Jeff Deemer, who some of you may know from having him as a TA, is also going to be at the biology graduation. Another plug for the biology graduation. I'll be officially conferring him his PhD, so he gets the funny hat to wear and all that kind of stuff. So that should be rather fun. Again, that's on the 13th. And what time is it on the 13th? Thank you. Noon on the 13th. So that being said, let's move on into the most important viruses of the whole term. Yes? Well, at least the ones that I think are the most interesting and important. And that's these guys up here, the Archaeal viruses. But there are also these kind of cool viruses that we talked about last time, which, of course, we have clicker questions about. So the most similar human virus to mimivirus is thought to be megavirus, smallpox, HIV-1, baculovirus, or hantavirus. Or should I say Hantan virus? Ten, 4, okay, which of these are human viruses? Smallpox, HIV, and Hantan virus. Baculovirus and megavirus don't infect humans. Okay, so HIV is a, what kind of virus? Retrovirus. Hantan virus, something that infects mice too. Also an RNA virus. So basically the only one that could even be close is smallpox. Also, it turns out that there are a number of genes that are present in all pox virus genomes that are also shared in all of these, what was it? Nuclear cytoplasmic large DNA viruses, NCLDVs. Again, horrible abbreviation, but that's, again, what they have. So entry by PBCV1, oops, let's actually give you, appears to be most similar to entry by adenovirus SV40, bacteriophage T7, bacteriophage T4, or influenza A. 3, 2, 1. Boom. So what do we think? C. Yes. We will talk about bacteriophage T4 on Wednesday after George talks about prions. So we'll be doing prions followed by bacteriophage T4 on Wednesday. So that's going to be the order there. And yes, it's like bacteriophage, and yeah, there's some differences between bacteriophage T7 and PBCV1. Yes, there always are. And I also overheard somebody saying, I hate Stedman's most like questions. I think that's a general feeling, not just me. So, but yes, C is correct. So it can't be that bad. 94% of you are getting them right. So can't be too horrible, right? What was the most surprising gene to be found in the mimivirus genome? Ribosomal RNA, tRNA, amino-acyl tRNA synthetase, RNA polymerase, DNA polymerase. So everybody yell out what they think is right. Yeah? Ten. Amino-acyl tRNA synthetase. So, hallelujah, 100%. Woohoo! So far, nobody's found the ribosomal RNA gene in virus genomes, but I would not bet anything against them finding one eventually, depending on how long it takes them. tRNA genes were kind of surprising to be found in virus genomes, but why were they not so surprising in the mimivirus genome? They've been found in other virus genomes already. Amino-acyl tRNA synthetases, this was the first one. RNA polymerases and DNA polymerases, dime a dozen. They're in all kinds of different viruses, right? So maybe I should actually select the correct answer now and make sure we all agree. So give people a chance to clear out if you guys need to take off. Unfortunately, week from Tuesday, you will not be able to discuss with your friends and neighbors what the answers are. Sorry. One day, eventually. Pardon? Final will be in this room, but at 8 a.m. on Tuesday. So really bizarre. But that's the standard exam time for the Monday, Wednesday, Friday at 9 a.m. So please be here on time. I will do my best to be here on time as well. So again, my apologies for those of you who thought we were going to be talking about T4 today. We'll be talking about that again on Wednesday. Instead, we'll spend the next 50 minutes or so talking about something that, again, we could hopefully spend a whole term talking about. These are the Archaeal viruses, and again, near and dear to my heart because I work on them in my lab. They are almost all double-stranded DNA viruses. Sort of a couple of take-home messages here, again, big picture-wise, is that the Euryarchaea, and we'll get back and talk a little bit more about Archaea and the different divisions in Archaea in just a minute, mostly look bacterial, i.e. head and tail type kinds of viruses, whereas the Archaea are bizarre. And this is just the beginnings of the bizarre in terms of the morphology, the genomes, etc. These things really do kind of look like viruses from outer space, at least Of course, we haven't had any of those yet. But they're just very, very different in terms of what those particular virions in their genomes look like, but they still adhere to some of the very similar principles to what we've been talking about in terms of a number of other viruses already. So again, many viruses of the Euryarchaea are like bacteriophage, and again, that's at the 30,000-foot level. Viruses of Archaea are way different, and then one of my favorites again up there, actually probably my favorite virus, the SSV virus, the Fuselloviruses. We're mostly going to be talking about those today for two reasons. One, I work on them, and two, because probably the most is known about them in terms of all of these different archaeal viruses. Then we'll just do a quick overview of a few more virus families, and basically virus families so far have been one chapter of the textbook, right? And now we're doing, actually I think there are about 10 virus families now in the Archaea that we're doing in one chapter in one lecture. So we'll just sort of screen over these relatively quickly. The Rudiviruses are rod-shaped viruses. The Lipothrixviruses, these long filamentous ones. Globulo, should be pretty obvious what they look like. Ampullas are bottle-shaped viruses. So actually before you had the Pandoras and the Pithos, these are the first ones that sort of look like liquid containers. And then the Bicotaviridae, again, for those of you who've had Latin, which I guess is not a huge number, two tails. So the two-tailed viruses. And these are really amazing because what happens is they grow their tails outside of the cell. And so this is extracellular maturation, which is just really crazy. And then we've talked a little bit about these Turiviruses, the STIV viruses. So yeah, we've got all these crazy number of virus families. Why do we only have one chapter in the textbook? That's because most virologists think like this. Remember our friends, the ICTV, or not friends as the case may be. They had this wonderful Virosphere of all the different viruses that are out there, double-stranded DNA, single-stranded DNA, positive strand, single-stranded RNA, negative strand, single-stranded RNA, double-stranded RNA, and then this tiny little segment over here for the Archaea, which is just not fair at all. But if you also look at this, the viruses of bacteria are all right here as well. And so this is clearly a highly biased approach to looking at virology. I think a much better way of looking at viruses is looking at the hosts, or thinking about the hosts. And this is basically the same tree that I showed way back when, at the beginning of the class, where if you look at the molecular relatedness of organisms, there's really three big groups: Archaea, bacteria, and eukarya. And 90-some%, probably about 95 or 96% of viruses that people study infect these two very molecularly closely related groups of organisms, which are plants and animals. And just by the length of these branches gives you an idea molecularly how similar they are to each other, how much diversity is there. There's clearly a whole bunch more diversity in the cellular biosphere that's out there, and my focus is really on the Archaea. A couple of reasons for that. One, they're everywhere. People used to think they were just present in extreme environments. Turns out that they're all over the place, in soils, in water, etc. One way that we always try and get money is to try and convince people that they're really similar to eukaryotes, which is not a big stretch because it turns out that their DNA polymerases, and most importantly their RNA polymerases and how their gene regulation works, is actually really similar to eukaryotic systems, but much simpler. They have one RNA polymerase instead of three RNA polymerases. They have TATA boxes, just like you'd expect for a normal transcription in eukaryotes, but again, very, very similar. They do thrive in extreme environments, and that's how I got interested in these guys. They're basically living in boiling acid. How on earth can you do that? Everything I learned in my molecular biology biochemistry courses says you can't do that, but they do. One of the reasons that they can survive under these extreme conditions is that they have these really fascinating tetraether lipids. So the ether lipids, in fact, quite similar to eukaryotic systems, as opposed to the ester lipids, which you have in most bacteria. But the tetraether lipids, which is we have a lipid monolayer on the outside of a lot of cells, as opposed to a lipid bilayer, which is probably partly how they can survive in some of these extreme environments. And they may be similar to ancestral organisms. This tree here from Norm Pace, admittedly a couple of years old now, if you look at where the Archaea are, they're pretty close to the last common ancestor, at least molecularly, if you believe a uniform molecular clock and all kinds of other caveats that go with this. We particularly work on Sulfolobus, and then, of course, we're over here. The real surprise that people had when they first started to work with Archaeal viruses, which was literally only about, getting back to 40 years ago now, is that these virions don't look like any other virion that anybody's looked at before, with the exception of some of these guys, which look a lot like what virus? So what particular bacteriophage? T7 is a little different. T7 has basically no tail. This one's got a long, flexible tail. Lambda, exactly. So this guy looks a little bit like Lambda. This one we've talked about quite a bit, the STIV with the bizarre triangulation number, but also these beautiful projections at the five-fold axes of symmetry. But then there are these other ones that are just really bizarre. Long filamentous viruses with these nano-sized claws at the end that do close down on pili. Whether that's important for entry is not entirely clear. These guys that we're going to spend a lot more time talking about, these kind of spherical viruses, which I think actually look a little bit like influenza, some of these paramyxoviruses, but they don't have any obvious internal structure to them. And then this guy, which I think is the most bizarre morphology of any virion that I've ever seen. These are the bottle-shaped viruses, Acidianus bottle-shaped viruses that do kind of look like a champagne bottle, but also has this ring of fibers at one end that also kind of looks like candles on a birthday cake. My daughter's birthday was a week ago on Monday, so I know all about birthday candles. And so this was a really amazing virion morphology, and at first people didn't really believe that that was going on there. So we'll start talking a little bit about these viruses of the Euryarchaeotes. Euryarchaea are one of the two main branches of Archaea. These were some of the first study because they were the methanogens, which were the first ones shown to be Archaea, and also some of the extreme halophiles. As I may have mentioned before, anytime you fly into the San Francisco Bay Area, particularly if you're landing at SFO, you'll see all these different colors of all the salt ponds that they're doing the evaporations in. Anytime that goes to purplish, again from red to purple, that's all Archaebacteria, about 90-some% of it. Most of those are these extreme halophiles, again, because they can grow at saturating salt conditions. So, again, not surprisingly, because these are some of the first discovered, also some of the first viruses were found. Most of these guys look, again, a lot like some of the other bacteriophage. We've got SIAM1 and Phi-H here, but then there are a few other ones. This one, if you squint at it enough, kind of looks like STIV. Turns out in cryo-EM it looks a lot more like that. But you also have a few of these spindle-shaped viruses. But for the most part, again, these viruses of Euryarchaea look very much like bacteriophage. But again, that may be because that's what people were looking for. And anytime anybody's done electron microscopy knows you see a bunch of junk in the electron microscope. And so unless you see a whole bunch of particles that kind of look like this or look like that up there for HIS1, at first your immediate assumption is going to be it's junk. And it's interesting, after I've given some of these talks before at some scientific meetings, people have come back to me and said, hey, I've seen stuff that looks like that in my EMs. I always ignored it. So that's another thing to think about here. But just as an example of some of these viruses and their similarities, particularly these Euryarchaeal viruses to bacterial viruses, Phi-H1 is again a virus which infects those extreme halophiles that you find in these hypersaline environments. The morphology looks almost identical to bacteriophage P2. That's because of this thick tail that comes off of the bottom here. And in fact, this is, you can't see it very well, but this is P2 and this is Phi-H. So they're really basically morphologically identical to each other. Curiously enough, however, the genome and the transcription are much more like Lambda for Phi-H1 than they are like P2, which we haven't really talked about, but it functions a lot more like bacteriophage T4. So the morphology is basically like P2, but the genome and transcription are like Lambda. It integrates normally. It seems to have a repressor gene, circularizes once it comes inside the cell. So there's lots of similarities between the two. So that's pretty much all I want to talk about in terms of viruses of Euryarchaea, and that's partly because that's about all that's known about viruses of Euryarchaea. So there's, I think, a lot more to be known about this, and people interested in what they want to study, I think this is a really cool field to move into. Instead, I wanted to spend the rest of the time talking about viruses of Sulfolobus. It's a good thing this is kind of washed out because you can't tell that my hair wasn't gray in this time. So this is me sampling in Yellowstone National Park, a typical kind of Sulfolobus habitat. This particular pool, it's 85 degrees Celsius, a pH of 3.5. Okay, it's nice you can't see me too well, but unfortunately, this really does look like tomato soup. This particular pool is really pretty amazing. And this is in the Rabbit Creek area. In fact, it was very close to here that I picked up the sample that I found STIV in. Not in fact this pool, but very nearby. So what do some of these viruses look like? I showed you some pictures before, but I want to concentrate on the Fuselloviruses. This is, again, mostly what our lab works with and my favorite viruses. The virions are these spindle or lemon-shaped particles. They're about 60 nanometers by 90 nanometers with a short tail at one end. We'll see that in a little bit better resolution a little bit later on. But what kind of symmetry does this have? Does it have helical symmetry? Does it have icosahedral symmetry? So, okay, what kind of symmetry does it have, and how do you get this assembly? And these guys are functioning at these very high temperatures and low pHs. So how can all these proteins be fitting together to do that? So, these are all unanswered questions at this point. That's what we're trying to figure out. Another thing which is interesting about these viruses is they have double-stranded circular DNA packaged inside these virions. This package is a positively supercoiled state. And that positive supercoiling probably is important for surviving at these really high temperatures, because positive supercoiling basically overwinds your DNA and keeps those two base pairs together, even at elevated temperatures. Yeah?
Is each individual cluster or each individual blob a virion, or is the entire assemblage the virion?
Right, so this here is one virion down here at the bottom. So one of these lemon-shaped or spindle-shaped particles, Fusello, comes from spindle. So it's just, this is one virion here. They love to bind to cellular debris, which is what this is, all of this stuff here in the middle. So that's basically what you're looking at. These are the pretty flowers, and people talk about them like sunflowers sometimes. I'm very proud of this EM because it's one of mine. In fact, I took this picture. So it is probably the best of about 300. So we're selecting from some of them. The other thing that this double-stranded circular DNA genome does is it integrates very specifically into the host genome. So again, very similar to something like Lambda or the parvoviruses. Also, like Lambda, you can induce production of the virions by irradiation with ultraviolet light. There's nothing like a repressor. There's nothing like LEX-A. There's no obvious SOS response. So how this happens is really very much up in the air and unclear. Yeah?
What did you say was similar with parvovirus?
Specific integration. So when it integrates into the genome, it integrates at a very specific place. And we'll talk about that in just a second. And this particular SSV, SSV1, was isolated from Sulfolobus shibatae from an onsen in Japan, in Kyushu, in the southernmost part. And I'll talk a bit more about why that's important a little bit later on. So, okay, you've got this virus. What's in that genome? Well, turns out that there are about 34 open reading frames. So it's got quite a few genes that are, or at least potential genes, we don't know if all of them encode proteins, in the genome. But we know very, very little about these genes. Basically, we know that these three proteins here, VP1, 3, and 2, not surprisingly, are the virus proteins. So these are the structural proteins. So when you isolate virus particles, that's what you find. VP1 and VP3 make up the majority of this capsid up here on the top. And VP2 is a DNA binding protein that binds nonspecifically to DNA, presumably is important for that positive superquilene packaging, although we don't absolutely know that yet. The only gene, if you look at all of these open reading frames, that shows any similarity to anything else in all of the databases is this integrase gene, which is, surprise, surprise, similar to lambda integrase, because that's exactly what it does. It takes the circular genome and integrates it into the host genome. It does that through this attachment site here, which is also, again, extremely similar to lambda bacteriophage. It's got a sequence in the viral genome that's identical to a sequence in the host genome. The virus integrase binds to both of these sequences and integrates the genome into that. A couple of things that are a little different. One is that that attachment site is in the integrase gene itself. So when you have integration, you in fact disrupt the integrase gene. And that probably means that that integrase gene itself is no longer functional. And so integration is probably a one-way street. Once you go in, you're always integrated. And that's in fact what we see. We see huge amounts of integration present in a lot of these genomes, and not just the Sulfolobus shibata one that this guy's originally from. In the middle here, I just wanted to mention this is a cryo-EM reconstruction from our collaborators at UT Medical Branch, where they've got that BSL-4 facility and the microscope that they can do at BSL-3. This is BSL-1. We can work with this in our lab, and people in the recombinant DNA lab as well. But one of the neat things about the cryo-electron microscopy is you can also see a structure of a tail down here. And this tail has six-fold symmetry, which is exactly like the symmetry of tail structures that you see in bacteriophage. And that symmetry matches exactly the coat of the Sulfolobus host, which has what's called a surface layer protein that's arranged in hexagonal symmetry. Hexagons fit nicely with this. So somehow there's some six-fold symmetry, which is here. But the connection to six-fold symmetry in this blob is still very much up in the air. What's going on with the whole rest of the genome? We basically have no clue. Or at least we didn't when I first got started on this far too many years ago. So one of the ways that we've been trying to figure this out is using a technique that we developed, and we, I mean my graduate student, Adam Clore, developed about 10 years ago now, which we call long inverse PCR. And basically the way that this works is you have two PCR primers that you design, but instead of facing towards each other, which is usually what you think about when you do PCR, they in fact face away from each other. But if you have a circular genome, this guys will now amplify that whole circular genome, and then if you just ligate those two primer ends back to each other, you generate a genome that's missing some gene that you're interested in. So we did this with the viral integrase gene, and we've been slowly going around the genome, again, we being Adam and Erik Iverson, who some of you know, in terms of trying to figure out what's going on in this whole genome, just by going through and doing some of these deletion processes. So again, you ligate those two ends together, you end up with a deletion of your favorite gene. In this case, we chose the integrase gene to knock out because it was the only gene that was similar to anything else in databases. And we said, hey, it's probably going to be important for function. We delete it. Turns out the virus is perfectly happy, which is kind of bizarre, and we're not quite sure why. It doesn't integrate normally. And the normal virus, actually, when you do a competition head-to-head between the two, the normal virus wins. But still, the fact that it's making viruses is still kind of surprising. To make, I guess, 4 years of Adam's thesis and about 3 years of Erik working in the lab's time really short, one slide. This is basically, or should say, these are the results of all of those deletion experiments, which both Erik and Adam have done. Basically, these are all the open reading frames in that SSV1 genome. Everything is green on here. You can knock out, and the virus is happy, which is really kind of surprising. You see there's a whole bunch of green all over here. So you can actually knock out a bunch of genes in the genome, and the virus seems to be quite happy, at least under lab conditions, under our particular lab host, and so that's probably part of it. A few genes here are red, and that's if we knock them out, then we don't have virus function. There may be all kinds of different reasons for that. We'll get back to that a little bit later on. I wanted to concentrate on those genes up the top. You remember VP1, VP3, and VP2. I'll start out with VP2. That's that DNA binding protein that we think is important for packaging, making positive supercoils. Well, you see up there it's green. We can knock it out. The virus seems to be perfectly happy. That's strange. Well, what happens if we knock out the gene right next to it, the VP3 protein, which is part of that capsid structure? When you knock that out, the virus seems to be perfectly happy. That's even more bizarre. And that we really don't understand, and we're trying to get some more data on trying to figure that out. Yeah? At the same time? No, these are separate deletions relative to each other. So we've done some multiple deletions, and in fact, down here at the bottom, we've done some deletions together. We've also tried to delete much larger parts of the genome, because you see over here, there are a bunch of green stuff that hopefully we should be able to get rid of the whole thing. Not been successful in doing that yet. Yeah?
I thought that PCR, you were limited in how big segments of DNA you could get. Isn't the SSV1 genome too big to do PCR?
Aha. So you may or may not remember, maybe I can back up a couple of slides here, that we're calling this long inverse PCR. So that is, in fact, it's a great question. And what we needed to do was come up with ways that we could do this very long PCR. And in this case, it's actually about 17 to 20 kilobase pairs to do with PCR. So it's really pretty long. And in fact, I gave a talk at a company called MJ Research, used to make thermal cyclers. They got bought by Biorad. And I was complaining that there was no way we could make mutants. And they said, we have this enzyme you should try called fusion polymerase. Some of you may or may not have heard about the fusion polymerase. It's a engineered DNA polymerase that's a fusion between two different proteins. It's a DNA polymerase from Pyrococcus furiosus attached to a single-stranded DNA binding protein from Sulfolobus. It is incredibly processive, so it stays on DNA for really long periods of time. And we beta tested that. In fact, that was part of the beta testing. And we got probably thousands of dollars worth of enzyme for free to beta test it to do this, which was pretty cool. But yeah, it's a great question. And in fact, it was really hard to do those kinds of long PCRs, but now Eric does them on a pretty regular basis. I'm not sure I would want to try. But he's really quite good at it. So done all these deletions, but it turns out that partly because that long PCR is actually kind of a pain to do, we said, well, there's still a bunch of gray things here that we don't know about. Well, Eric could go through and make all these deletions. Yeah, he's getting pretty good at it, but it still takes, on good days, about a month, or I should say good times, about a month to do any one of these deletions. And 35, you can do the math, how many months it's going to be, and he actually wants to graduate sometime. So we said there's another way that we can do similar kinds of things, and that is to do insertional mutagenesis. So we did insertional mutagenesis, but the problem with insertional mutagenesis is you end up with so many mutants, and each of these arrows represents one insertional mutant, that it's really hard to screen all of them. So you need extra hands. Where do you get extra hands? Undergrads. Exactly. So we've been using our collaborators in the recombinant DNA techniques lab to screen some of these mutants. In fact, basically what happens, and this is great practice for those of you who are going to be giving your talks in the recombinant DNA lab, say in about a week. Yep, a couple of days. Basically, what happens is you get an insertion, but you don't know where that insertion is. You have an insertion somewhere in the genome. You figure out where it is by cutting with restriction endonucleases, which will give you a shift relative to your wild type without an insertion. And then you can identify exactly where it is by doing DNA sequencing. Then, of course, you know where this arrow is, but you don't know if it's green or red. And so that green or red process is this functional as a virus or is it non-functional? And we'll talk about how we do that screening, we being, of course, all these other people, not me, who have done some of that. And I also do need to mention David Goodman, another undergraduate who's been working in my lab, and Virology Wong, who was a high school student who was working in my lab last summer, who's done some of this. We've now got insertions or deletions in every single open reading frame. This is slightly out of date. We do have insertions in this open reading frame and also up in some of the open reading frames up at the top there. So we've basically completely saturated the genome. A couple of interesting things come out of this. One is that, again, not surprisingly, where we can be able to delete a bunch of genes, that we can do insertions here as well. There are a couple of places down here in particular where you can't do insertions, but you can do deletions. We think this is because this is the origin of replication. What I didn't talk about is replication, as far as we know for these viruses, is theta replication occurring from one origin, probably down here at the bottom of the genome. So we think these insertions are probably messing up replication, as opposed to these smaller deletions, which we can do, that don't seem to be messing up replication per se. So there's also some strange insertions here that we don't quite understand, and we're still working on trying to figure those out. So we've done deletions, we've done insertions. There's one other way to do this, and this is actually a really fun way to go about trying to figure out what each of these open reading frames is doing, and that is to let the viruses and millions of years of evolution tell you about which of these genes are probably important or not important. So turns out that you find these SSV viruses pretty much anywhere where the conditions are right for Sulfolobus, so high temperature, low pH conditions. This is in Iceland where they found some isolates. This is near to a place in Yellowstone where I found some of these isolates. This is way too cold. It's only about 60 degrees Celsius for Sulfolobus here. And this is in Kamchatka in far eastern Russia, where I collected a number of samples and found some viruses there. Those pictures are way nicer than the map, but basically here gives you a couple of ideas where we found these from. SSV1, again, as I mentioned, from the southern part of Japan. Number of different viruses from Kamchatka, a few from Iceland, some from Yellowstone, and some from our favorite hunting ground down in Lassen Volcanic National Park. You'll notice they're all northern hemisphere. That's mostly because that's where we've been able to hunt. I think it'd be really fascinating to look at some of the hot springs here in the southern hemisphere, and particularly in New Zealand in terms of doing this. But the reason that we went there, other than being really cool to be able to go places and call it work, is that what we wanted to do was look and see which of the genes in these genomes were conserved, i.e. you find them in all these different viruses. Those are the ones that are likely to be important for virus function, and those that are not conserved, i.e. you don't find them in these other genomes, those are going to be the ones that are less important for function. And one of the really nice things that we found is when we did these genome comparisons, we found basically exactly the same results as our insertions and deletions. So you can delete genes that are not well conserved, the virus is happy, and you can't delete some genes that are really well conserved. Curious enough, the integrase gene is really well conserved. The VP3 gene is really well conserved, but you can still delete them. So it gives you some ideas, but not completely understandable. The other thing is that this tells us about virus distribution that unfortunately is a little beyond the scope of this course. I'd love to talk to people about it a little bit later. So basically, to make lots of samples really short here, this is in fact the SSV down here at the bottom from Yellowstone. This is that SSV from Kamchatka up here on the right. You'll see this one has a slightly different form to it. We're not quite sure why that is, but that may be because the genome's a little bit bigger for this particular virus. And these guys, one of the things you hopefully can see here is that a number of the virions are much larger than some of the other ones. Again, quite why that is not entirely clear. These virions seem to be pretty pleomorphic in terms of their structure. But in terms of talking about their genomes, basically about half of the genome is pretty well conserved, i.e. you find that half of the genome, it's about the top half of the genome, the way you've looked at it before, that seems to be really well conserved, probably essential, but we also know that there are quite a few that you only find in a few. And so some genes are not essential. We know this from the comparative genomics, but also from those insertion and deletion experiments I just talked to you about. If you look at the SSVs that we've actually been able to bring back into the lab and look at them, there's a really interesting correlation between how far apart any of the samples were that you collected and how similar they are to each other genomically. And so if you have, for instance, two viruses from Iceland, they're much more similar to each other in sequences than a virus from Iceland and a virus from Kamchatka, which kind of makes sense. That's exactly what you see in other kinds of cellular organisms as well. But that immediately brought up the question about host range. One of the things I haven't told you about is what these guys infect. And so told you Sulfolobus, SSV1s from Sulfolobus shibati, but we made an immediate hypothesis that these guys that are from one place, say Iceland, are probably going to be better at infecting isolates from Iceland than they are, and this is hosts from Iceland, than they are hosts from Kamchatka, for instance. And vice versa. Viruses from Kamchatka are probably not terribly good at infecting hosts from Iceland. Or you could actually have the exact flip side. Like you think about pandemic disease. Things that are transferring from one host to another. Think about MERS, from the camels going to humans. So something like that. Okay, so it's maybe a virus from Kamchatka is really good at infecting the hosts from Iceland, but actually pretty crummy at infecting other isolates from Kamchatka. So how do you go about doing these experiments? This is exactly the same way that our helpers in the undergraduate lab are doing this. Basically, we just look at, does that particular virus make that particular host sick? And the way we do this is a really modified plaque assay. We mix some of our Sulfolobus culture together with molten gellrite. Augury doesn't do very well at 80 degrees Celsius. So we put them on plates. We spot various different, in this case, more different constructs that we're looking at. We let the plates grow, and if the Sulfolobus is happy, that means we don't have functional virus. If the Sulfolobus is unhappy, that means that we have functional virus. So we've done this for lots of different isolates. And again, to make long story short, this is Caleb Marceau's honors thesis working in my lab. Remember those two predictions we had? Viruses from one place either are going to infect local hosts really well or different hosts really well. Well, it turns out neither is really true. So if you look at various Italian isolates here, so basically the way this is set up is all the hosts are the different rows, and they're color-coded relative to where they're from. And then the columns are different viruses, again, color-coded as to where they're from. So SSV1, that virus that we're looking at, was originally isolated from Japan. Turns out doesn't infect any of these Japanese isolates. Okay, so turns out SSV1 is actually kind of a wimpy virus. It only infects a couple of Italian isolates. On the other hand, this SSV that we isolated from Kamchatka infects everybody with the exception of one Japanese isolate. So basically, both of our hypotheses are kind of out the window. It's neither geography of one determines its host range or the host range determines what the susceptibility is. So we still need to try and figure out what's going on there. Another nice example of these two isolates from Iceland, SSV2 and SSV3, which infect some Icelandic ones but not other Icelandic ones, and different Kamchatka ones, etc. So there's still a lot to do to try and figure out what's going on here. But we have a genetic system, so we can actually start to do some of these experiments. And in fact, that's what some of the people in my lab are doing right now. So review of these Fuselloviruses. They've got an integrase, which actually they don't need. They've got lots of genes in their genome that don't look like any other genes in databases other than some other SSV viruses. But it turns out a lot of them we don't need. You find these all over the place. There's no correlation whatsoever. Kind of a bummer, that was our hypothesis, between host range and where these guys are actually from. And this SSV from Kamchatka, the one that's slightly elongated, seems to be infecting more or less everybody. And quite why that is, we're trying to figure out. And actually, that's what the 431 lab is doing right now as we speak. Or at least they'll be talking about it on Wednesday. So if anyone's got some time on Wednesday afternoon, come and hear what some of your colleagues have been doing in terms of doing some mutagenesis in SSVK. So last 20 minutes or so, wanted to zoom through some of the other viruses of Archaea that we understand a lot less about. So I can't give this whole spiel about them, plus we don't work on them in my lab. But the Lipothrixviruses, so lipo being fat, or basically membrane-coated thrix filamentous. These are amazingly long virions. This down here is a 100 nanometer scale bar. These are about 2 microns in length, the whole virion itself, which is even more surprising if you think about the host. The Sulfolobus is only about a micron in diameter. So these virions are about twice as long as the cell that they're infecting. Now again, how you deal with this in terms of assembly, packaging, you name it, is really pretty amazing. And again, nobody really has a very good clue. They also have these fascinating structures at the ends of their virions. In the case of AFV, it's these claw-shaped structures, but it turns out there are a number of other virions of this same family that have different terminal structures. So that variation in terminal structures seems to be the big difference between all of these different ones. Look at their genomes. They're double-stranded DNA with covalently closed circular ends. So this is really similar to what you have on PBCV1, number of other viruses. So you can either think of these as single-stranded, completely complementary genomes, or a double-stranded with covalently closed ends. This is just basically 6 of one, half a dozen of the other. Particularly in the case of these Lipothrixviruses, they've had these interesting repeat sequences at their ends, as well as an inverted terminal repeat, which is probably important for its replication. Inverted terminal repeats should sound very familiar to what we talked about in a number of other viruses. Again, it's the same sequence on the opposite strands, but then repeated sequences in those that actually look a lot like telomeres. There's nothing that looks like a telomerase in here, but all these repeated sequences that you find at the ends. And these are just some of the references here to some of those papers. The ABV, again, this has just an amazing structure. How it gets put together, unfortunately, we really don't have a very good idea. They're big virions. They're about 200 nanometers in length, a little over 200 nanometers, almost 100 nanometers in width, and have this, again, really amazing structure with these filament-like, almost, again, candle-like structures. They're 20, and they're always 20. How do you get 20? 20 is a really bizarre number. 20 faces makes sense if you think about an icosahedron, but how you get 20 of these in a circle? Who knows? They have a linear double-stranded DNA genome with protein prime, so that should sound a lot like what kind of virus? Adenovirus, exactly. So linear DNA genome, protein primed. The DNA polymerase, not surprisingly, people are interested in because these guys also are replicating at high temperature and low pH. So you automatically have a DNA polymerase that's working at high temperatures. So this, in fact, I believe has been patented in terms of trying to maybe use this. One of the big questions, of course, with this is how does it get into cells? How does it get assembled in cells? Unfortunately, it turns out it's really hard to work with these guys. You get very, very low titers. And these are, I think, the only pictures that I know of, EM anyway, that have more than one particle on them. Very challenging to work with. But here you can see really nicely on B here, we've got those filaments at either end. Those seem to be attaching to each other, but in some cases you see what looks like the cork end of the bottle associated with each other. So how these guys are entering into cells, how they're being replicated is really not obvious at all. And again, unfortunately, they're very, very low titer, so it's hard to work with these guys. Although I just saw a couple of EMs a couple of weeks ago from some samples from Yellowstone. These are originally from Iceland that looked like it may be a way to deal with this. We may actually be able to get a lot more of these viruses from a slightly different source. So I'm pretty excited about being able to go ahead and follow up on that. You also have viruses that, again, look like some of these enveloped eukaryotic viruses, the paramyxos, the orthomyxos, etc. This particular one is from Pyrobaculum. What's curious is that if you look at how its genome is organized, all except for 2 of the open reading frames are all on one strand, even though it's about a 30 kb genome. So it's really bizarre that they're all encoded on one strand in one direction versus another direction. Probably has to do with replication and transcription, because again, these guys have the same problem. They still have to replicate in the same way and make their proteins in the same way as all of these other proteins, all the other viruses we talked about. So if you have the polymerases, RNA polymerase and DNA polymerase going in the same direction, that would kind of make sense because they're not ever going to be running into each other. So that's total hand-waving at this point. We just know about the genome, what it looks like, linear double-stranded DNA, ITRs, and then it turns out there are quite a few of these viruses also these kind of pleomorphic shapes. There's a thermoproteus virus and this pyrobaculum virus as well. Yeah?
Is it known if the ITRs in this Globuloviruses and Lipothrixviruses, whatever you say that, does it make the frying pan structure?
Oh, in terms of how they're replicating? So, yeah, the question is just, do these ITRs make firing pans a lot like adenovirus? Because, of course, adenovirus also has these ITRs. And so the question, basically, I'm going to flip it around a little bit, and that's how do these guys replicate? And the answer is, we really don't know in most cases. The best studied are, in fact, the Rudiviruses, which I don't think I have any slides on here, but they're rod-shaped viruses that are infecting also Sulfolobus, originally found in Iceland. And there what seems to be happening is replication seems to be happening through sort of a homologous recombination type mechanism rather than the frying pan mechanism. But again, that's still very much up in the air, and people are talking about how that happens. But probably what happens, you end up with two genomes replicating right next to each other, and then you have recombination, which takes the repeats that you have between the two and then generates a single genome out of that. But it's, at this point, still very convoluted in terms of how people think is actually happening. Because if you think about the genomes here, again, if you've got a continuous single strand that loops around, comes back on the other strand, how do you replicate that and end up with a structure that also has these covalently closed ends? And the easiest way to think about that is actually something like what happens with parvoviruses, where you make a nick and use that as your starting point. But then where do you stop? The parvoviruses are single-stranded, so there's an obvious place to stop. But these are all continuous. So potentially you could just go around and around and around and around and around, and then you end up with multiple copies, and those then are acted on by the recombination enzymes to give you individual genomes. But yeah, the answer is basically we don't know, but I can hand wave. Oh, I do have Rudiviruses here. Sorry, I'd forgotten about that. But basically it's the same thing. Oops. I can't complete my backup. Yes, I know I can't complete my backup because I don't have a hard drive attached to this machine. So this is, I think, for most people, the most exciting of these Archaeal viruses. It's one of the very few that actually got published in Nature. And this is the one that I talked about right at the beginning. This is the B. Caudaviridae, so two-tailed viruses. These are really interesting because when they're produced by the cell, and this is Acidianus, by the way, is a very close relative of Sulfolobus. You also find them in the same kind of environments, high temperature, low pH environments. They grow under slightly different conditions. They're more autotrophs than they are heterotrophs. But here they produce these, nice, friendly, Fusellovirus-looking spindles. But then if you incubate these spindles in the outside of the cell at 70 degrees Celsius, what they do is they start to grow these tails at both ends. And then once they've grown these two tails, now they're infectious. The tailless form is not infectious. So when these guys are produced, they're not infectious particles, but they undergo a maturation extracellularly, which then allows them to be infectious later on. And so this is really, I think, probably the most amazing case that I know of any of these viruses having extracellular maturation. We did see extracellular maturation for HIV because the protease in HIV will cleave up the polyprotein in order to reassemble that nucleocapsid on the inside, but nothing like this. And this, at first, people were like, well, how the heck could this be going on? Viruses can't metabolize, right? So how can they be growing something? Well, what happens, and if you look carefully, you'll notice that these guys are a little plumper than these guys over here. So all that it is, in fact, it's just a conformational change. There's no change in volume between these particles and these particles. All that's happened is there's been an internal change in the structure. And so basically, this process here, sorry, it's a little bit hard to see, but at 75 degrees, you go from these spindle-shaped particles to these elongated particles that also have these hook-like structures at the end. They don't have the hooks when they're in this form. They do have the hooks here, and that's probably important then for getting the infection process to take place. These guys can both function lytic or lysogenic, very large genomes, and also have IS elements, and here's the reference here. Rudiviruses, so I thought that I'd skip this slide because it's put it in a slightly different order now. These guys also have these covalently closed hairpin ends, long internal repeat structures. What's most interesting, I think, about these particles is that they're non-enveloped. So we had the Globuloviruses are enveloped, the Lipothrixviruses are enveloped. These guys just have protein on the outside. This should look really familiar. What's it look like? Tobacco mosaic virus, exactly, except this is now a DNA virus, double-stranded DNA virus. But just like tobacco mosaic virus, the virion itself is just as long as the genome itself. And so it's packaged based on exactly how long that genome is. And so one of the things that some of my collaborators have found is that different length genomes give you different length virions. Do you have a question?
Yeah, what are IS elements?
IS elements, insertion elements, basically transposons. So in fact, what we're using in the Bi 431 lab to make all of these mutants with. Insertion elements are transposons that are jumping around. One of the things that was found in that ATV genome is that there are these transposons that are jumping around in them. Again, replication here, these covalently closed ends are a little bit like the, excuse me, the parvoviruses. We didn't talk about poxvirus replication, but it turns out poxviruses have very similar covalently closed ends, so these hairpin loops at the ends that are probably replicating again in a very similar way like the parvoviruses, even though the poxviruses are ginormous genomes. They're hundreds of kilobase pairs in size. But again, probably replicating in a very similar way. These guys also have some fascinating tail-like structures at their ends, which you can also see quite nicely in the EM. We found a number of new virus-like particles in Yellowstone. In fact, that picture of the tomato soup spring is very close to Rabbit Creek, where I found STIV. We also have some viruses that look a little bit like that two-tailed virus, and also some really bizarre-looking things that we found over there. I'm going to skip over STIV because we've in fact talked about this already in terms of the really cool structure that we think means that this is a really old virus. Just to finish up by talking about some virus exit mechanisms, which are really unique to some of these archaeal viruses. And this is, again, I think one of the really fascinating things to come out of the study of archaeal viruses. It's a whole new mechanism that seems to be unique to the archaeal viruses about how viruses, in fact, get released. So if you look at an SEM, this is a scanning electron microscope image of cells that are infected with either STIV or SIRV, which curiously enough are not related to each other at all. Remember, SIRV is this rod-shaped virus, looks like TMV. STIV has that nice icosahedral structure. Both of them, when you infect cells with them, produce these pyramid-like structures on the outside of the cell. And then after a certain period of time, these pyramid-like structures open up like petals on the leaf of a flower, and the virus is released through them. And so these are some recently published, sorry, I should put the link on here, I'll do this on D2L. But here's what looks like a virus particle that's almost ready to be released underneath one of these pyramid structures. It opens up, the virus particle is released. So basically they're making these holes in the host membrane and then also the proteins that are present on the outside. One thing that's really interesting about these pyramids is that they're seven-fold symmetric. How the heck do you get a seven-fold symmetry in a biological system? Nobody really knows. But they, and the process of how they open is also not clear. It's probably dependent on the number of virions that you have inside the cell. Yeah?
So is it just one virion per pyramid?
That's a great question. So the question is it one virion per pyramid? It's not clear whether it's one virion per pyramid. For STIV, it might be. For SIRV, it seems to be multiple virions that are getting released per pyramid/hole. Yeah?
So is this viral proteins that make up that structure, or is it manipulation of
Great questions. The question is it a viral protein, is it a cellular protein? If you look at the two virus genomes that I said are really very different from each other, the rod-shaped genome and this icosahedral virus genome, they have one and only one gene that's similar between the two. And that particular gene is the one that makes these pyramids. And if you express that one protein in Sulfolobus, it makes these pyramids. If you express that one protein in E. coli, it makes these pyramids. You express that one protein in Saccharomyces cerevisiae, it makes these pyramids, which is really, I think, kind of mind-blowing. That just one protein, one viral protein, you make it. The other thing which is really interesting is that not all of the viruses, even the STIVs or the SIRVs, have this particular protein. Also very strange. Yeah?
So is the viral protein the actual structural protein that makes the pyramid, or is it just something
that influences pyramid formation? So the question is it just that viral protein that makes the pyramid, or is it something that induces pyramid formation? It is the viral protein. And so we know that just if you do some immuno-EM, etc. So to finish up, totally cool number of viruses. This, I think, is a much better way of thinking about virions than that ICTV structure here. I don't expect you to read the names here, but basically 90% of that thing at the beginning were these guys, but obviously we've got a lot more diversity in the Archaea and the bacteria, red and blue here. This is just basically what we talked about already. Most importantly, I need to mention David Prangishvili, whose lab did basically all the work other than the Fusellovirus work that I talked about. He's at the Institut Pasteur in Paris, although for about 3 years we sat right next to each other, the same bench at the Max Planck Institute for Biochemistry in Germany, in Wolfram Zillig's lab, again, mostly David Prangishvili. And then just as a quick plug for Lassen Volcanic National Park, this is one of our sampling sites down there. So we'll see you on Wednesday for prions and bacteriophage T4.